All right, so now we've kind of talked about more kind of local molecules as well as cell adhesion molecules. And now we can talk about more local and long range guidance molecules in terms of things that are more diffusible. So that's where we're kind of going here. Most of our guidance ultimately boils down to either repulsion or attraction. I know we've kind of mentioned this further, but it's a good thing to kind of hit on again. So, outgrowing growth cone may be attracted to a specific molecule, so that is going to cause it to create Philopodia that will move in that direction, and then ultimately kind of turn or navigate that way, or they may have a repulsion, which will make them turn away. And as mentioned previously, most of the categories of molecules that do this are either ephrins, netrins, semaphorins, or slit molecules. And remember that whether they serve as attractive or repulsive depends on the cell receiving the signal and the time at which they receive it and the receptor that they have. So it's not like ephrins are either always attractive or always repulsive. It depends on the cell that's receiving the signal. Okay. So let's kind of look at an example of this. So one example in the developing limb is actually moving a basically sensory neuron if the cell body is out in the periphery and then growing back into the central nervous system. So this TI1 sensory neuron here needs to go back to the central nervous system. And so one of the things that we can do is as it's growing back towards that central nervous system, we can then put up a band of semaphorin 1. Now, semaphorin 1 in this instance is a repulsory cue. And so it's not going to try to cross that semaphorin 1 up here. Instead, it's going to turn and move in either this kind of positive or negative direction in that developing limb. Now it will travel until it starts to reach a gradient of CX1. So CX1 is a diffusible attractant that actually overrides semaphorin 1 expression. So as we travel down here, that CX1 becomes way too appealing. And so it's going to move over and actually finally cross semaphorin 1. And then it can attach into the ventral nervous cord, the ventral side. So this kind of idea is that we set up this boundary. However, at one point in time, something becomes more attractive, allowing that axon to cross. Now, interestingly enough, we do have semaphorin 1 knockouts. And if we are missing this kind of semaphorin 1 band, what ultimately happens is we get this random searching behavior. So what you'll see is that if this neuron got to this point and then had this semaphorin 1 band, you see it might go up, back this way. Oh, maybe I'll go this way. Oh, here's CX1 and it goes this way. So you kind of get this random meandering that ultimately occurs with knockout in that area. Okay. Another example of this is what's called semaphorin 3 or collapsin. And semaphorin 3 and collapsin will either be excitatory or inhibitory depending on the type of neuron that it runs into. So if we look at neurons that have their cell bodies in dorsal root ganglion, these dorsal root ganglion neurons then will innervate different positions within the spinal cord. So neurons that are responsive to NT3, meaning that NT3 is an attractive cue. This is semaphorin responsive neurons, is NT3 responsive neurons. They will actually move down and go here into the ventral portion of the spinal cord. You can also have those that have low threshold mechanoreceptors. They are not as attracted to semaphorin, and so they're going to stay more up here in this more dorsal horn of the spinal cord. And then temperature and pain receptors, they do not like semaphorin at all, so they actually end up staying as far away as possible up here at the top. So if we kind of take this example and we take it out into a petri dish, we can have a early kind of neurons. So early neurons, they send out kind of signals all over the place. So they actually look more like these big kind of puffballs. Then they actually look like neural cells. And then if we place a semaphorin 3 secreting cell next to it, If this early neuron has mechanoreceptors, it is set up to be a mechanoneuron. We see a repulsion, meaning that we collapse back this whole side here and we stabilize protrusions out on the opposite side. However, these neurons that have NT3 receptors within them that would ultimately go into this more ventral portion of the neural tube, they are attracted to semaphorin, and you can see that they've actually kind of sent out these projections here that are interacting with that semaphorin secreting cell. All right, now these are kind of some static examples. Looking at these more in kind of a active motion is really cool. So this link, I cannot get these YouTube videos to play on my iPad when I'm doing this recording. I'm not sure why. I've actually put this link into the modules tab. It's also stuck here within the recording. If you want to go ahead and search that out. That way you can kind of see these guys actually in action. So what it is it's a recording of a bunch of different growth cones using a bunch of different factors, and you can actually see them sensing and then turning. So it's a good video to watch. All right, and then last but not least, we have kind of the final step, which is this address selection, or a lot of times what we call synapse formation. And this is basically once we have finally made it to our target and made contact, we've made it to that address, how do we then form a synapse? And this process takes the form of many steps. So kind of the first thing that's going to happen is, for instance, this is a motor neuron that is growing out to innervate muscle. And so when the growth cone first contacts the early myotube, or what is going to become muscle, it will basically kind of search around and create kind of these multiple contacts that are non-specific contacts. Now, those unspecialized contacts then become specialized contacts through a couple of different processes. The first is that we produce what is called agrin, the muscle cell itself does, and this agrin on the surface of that muscle cell ultimately help with that contact formation. Through interactions with that agrin, we then start to create things like neurotransmitter containing vesicles. In this instance, it's going to be in acetylcholine. So we start to create those vesicles. This also induces the extracellular matrix of the neuron to interact with B2 laminin. This is a adhesion type molecule. So we're kind of creating an adhesion between the muscle cell as well as the outgrowing neuron. This then also induces clustering of acetylcholine receptors. So we create acetylcholine receptors on behalf of the muscle cell right underneath that kind of early neural tube. So we start to set up kind of this preliminary structure. However, formation of this nerve into the muscle is so important. We also send out signals that then attract other axon components. So this outgrowing cell now is going to create multiple different axons that will then interact with this area. Now, once we have these multiple different axons, we will then have them compete to see who can kind of create the best interaction overall with these areas. So we go from this instance here where we have multiple axons innervating, and then the strongest one is ultimately the one that is selected and kept. That leads to that axon then expanding out, taking over more area, and then we wrap it up with Schwann cells, creating that myelin sheath, which creates our kind of full-on axon interaction. So in order to make sure that we create these connections, we will send out multiple axons into that area, and then whichever one attaches the best is the one we keep. The rest we prune back and get rid of. Now, this process of pruning back is a really good example of programmed cell death, or apoptosis. In the developing nervous system, more than half of all the neurons that we create actually die. And this helps us to make sure that we create the connections that we need to where we need to. It ensures proper spacing and orientations of neurons. In other parts of the body, it will actually help to generate inner ear space. vaginal opening in spaces between fingers and toes, and we also pruned back unneeded structures. It can also help us to kind of sculpt specific tissues and complex organs. We'll talk about this more when it comes to, for instance, the developing limb. This actually helps us to sculpt like the bones and the muscles specifically within the developing limb. Now, we know from work in C. elegans as well as in mammalian models such as mice that there are a group of proteins called SEDs that seem to be fairly important in this process. So SED9 is a blocker of SED4 and SED3, which then trigger apoptosis. So if we remove SED9, ultimately then that allows SED4 and SED3 to be activated, leading to apoptosis or pruning back of those tissues. Now, in mammalian neurons, we kind of have some corresponding factors to this. SED9 is actually called BCL2 in mammalian neurons. That's why these boxes are colored ultimately the same way. Now, SED4 is APAF1, and then we have, in terms of SED3, we have caspase-9 and caspase-3. And these then drive apoptosis. So the presence of BCL2 stops that, whereas APAF1, like SED4, will then drive this process. And we can actually see this, some of the problems that occur with this. So if we look at a wild type developing mouse, and here we can see the blood vessels, as well as kind of the brain, because the mouse is relatively kind of transparent at this point. He's small enough. However, if we knock out caspase-9, which usually is important for driving apoptosis, right? So it's like we get rid of this step in the chain, and so we can't activate caspase-3, leading to apoptosis. We see that we have kind of this enlarged brain. The vasculature is a lot more complex here. We have way more vasculature than we need, and this ultimately leads to developmental defects. So we actually need this cell death as an important part of this overall process. All right, guys, that is it for